My partner, Shantae Fyffe, and this summer we interned in a microbiology lab in Brooklyn College with our mentor, Peter Lippy, and the title of our presentation is the growth of these biofilms, and that's a picture of Brooklyn College right there. Okay, let's do some background information. Okay, basically, in our lab that we're working with, that we intern in, we work with Saccharomyces cerevisiae, and Saccharomyces cerevisiae is a species of yeast cell that is commonly known as baker's yeast, and baker's yeast is like bread, things that we use in bread and wine and other types of things. And in the lab, we're working with ALS5, and ALS5 is a mutant of Saccharomyces cerevisiae, and it's also a gene from Canada albicans.
ALS5 is both a gene and a protein. It's a protein from Saccharomyces cerevisiae, and it's a gene from Canada albicans. The way to tell the difference between ALS5, the gene, and the protein is that when it's written, the protein is written in lowercase letters except the A, it's capitalized and it's italicized. And then the gene, it's just written in all capitalized letters. There are 8 different strands of ALS genes. There's ALS1 through ALS7, and then there's ALS9.
Okay, so right there, that's thrush, which is a type of yeast infection. Okay, Canada albicans is a fungus that causes yeast infection, and it affects maybe 10 out of 1,000 people a year, and it kills hundreds of AIDS victims a year, and it only attacks people with suppressed immune systems, which means it's an opportunistic pathogen. Biofilm. What is a biofilm? Basically, a biofilm are communities of bacteria or other single-cell organisms organized in slime. Basically, when we express our ALS5 gene on our agar, which is going to be 0.3% agar, we want them to look like biofilms, which are slimy, shiny. They should be cream, irregular, and large, which is going to show growth. Okay. And examples of biofilms are soap scum. Your mouth has biofilm. If you ever touch water, if you go to the beach, you touch the rocks under the water, and the rocks are slimy, that's biofilm. So yeah, you guys don't have to copy all this stuff. And our agenda.
We want to discover if ALS5 changes the way yeast grows, and what is the difference between the way yeast grows on ALS5 and on the other proteins, which was PADH, ALS1, ALS3, and the wild type W3O31B.
Yeast cell. This is yeast cell that I drew last year. This is the nucleus, and this is the plasmid. And basically, in the plasmid, there's a promoter and there's a gene. The gene encodes what our yeast cell should be expressing on, and the promoter is basically going to tell the rest of the cell that this is what you should be expressed on. You should express on 0.3% agar. Yeah, so that's what that shows. Materials.
To do our project, we use petri dishes, flask, an incubator, an autoclave, YPA agar media, CSM-0 agar media, CSM complete agar media, and the strains were W3O31B, ALS5, ALS1, ALS3, and PADH. We use a pipette, a shaker, gloves, CSM-0 liquid, CSM complete liquid, a spectrophotometer, and a fridge.
All right, step 1. We use 3 different types of, this is our methodology. We use 3 different types of of medias, and one was YPA. The other 2 were CMS-0 and CMS complete. And basically, there were 4 flasks of each of these medias. Okay, and as you see here, 2 of them are 2% agar, and with galactose, and one with glucose, and other one is 0.3% agar. And the difference between 0.2% and 0.3% is that when we pour them on the plates, the 0.3% are semi-solid. So they're not. So the 2% are more solid than the 0.3%. And basically, we had 12 flasks
from the air or that was inside the flask would be gone.
All right, step 3. After we took them out of autoclave, this is a picture of us pouring. We pour from the flask, we poured it into the plates, and then we let them cool. We let them cool for about a day, and we just it was more. This is just maybe half of them, the plates that we made.
And then after the plates were cooled, we had to seal them with Biofilm because we didn't want any air to get inside of them and contaminate our results. So we sealed them and put them in the fridge. The 2% were placed upside down because we didn't want the water to get on top of it. But the 3%, they were placed the right side up because they were too fragile. And if we turned them upside down, they would just break. So we would have to do it over again.
And step 5, this is this is going to be a video of us inoculating. We made a suspension media. We made 5 different flasks of white PS suspension media, and each flask had a different strain: white PADH, ALS1, ALS3, ALS5, and W303-1B, which was our wild type. And this is how we inoculated it. And that's the video of us taking the strains from a petri dish and putting it inside of the media. And then after that was done, we put it into the incubator shaker.
The strains were already made, so we just took them from the petri dishes and put them in the suspension.
Yeah, thanks, Janice.
This was a shaker. It was in the shaker for about 2 days. The first day we took them out, it wasn't grown properly, so we had to put it back inside. It was put at 25 degrees Celsius, shaken at 175 revolutions per minute.
All right. When they were ready, we used a spectrophotometer, which measures the optical density, which is going to show us the cell concentration and the cell concentration of the YPS suspension medias. And these are the different strains. These are their ODs. ALS1 was 1.85, ALS3 was 1.38, and so on. So they were all about the same, except for wild type, which is different, W303.
This is showing when we inoculated the strains on the petri dishes. When the suspension came out the shaker, we used a pipette, a 10 microliter pipette, and inoculated one of each strain on each of the petri dishes. So we had 5 petri dishes for each media.
Okay, step 9. After we inoculated all the strains on inside all the petri dishes, we placed them in the fridge in the incubator, which is 30 degrees Celsius, which is maybe, I don't know, that's warm.
When the YPA suspensions came out the shaker, they smell like beer. They smell like beer because beer is made from yeast.
And while we were conducting our experiment in the lab, we had to be very sterile because there's a lot of bacteria in the air. Even though you guys can't see them, if they go on our agar plates, they will contaminate them and there'll be fungus, and that will alter our results. And it's 0.3% with glucose, and this is CMS, and this is how this one looks like. This is not what we want to look like, but this is a different strain, I guess. And this is 0.3% with galactose. Yeah.
At the end, they're all supposed to be white, so.
Okay, this is what we would like to look like. This is kind of small right now, but you see that it's cream. We couldn't take the covers off because they would get contaminated. But it's cream, it's shiny, and it's slimy, and it's irregular. So maybe in about by Monday they should be bigger and showing growth. And this is 2%. You can see the 2% are more darker than the 0.3% because this is more solid than this one. And these are some of them in the fridge. So they all look different because they're different types of the strains, and they're all on different types of media, so they will be going differently. And we'd like to acknowledge our Professor Lipke, Peter Lipke, Crystal Strauss, the Lipke Lab, and Harlem Children's Society staff, Dr. Sat Bhattacharya, and Brooklyn College.